Yeah, good morning, good afternoon, and good evening. So we are here today that climate ready classroom, the public health faculty development course on climate change. So we are today in our climate change and vector-borne disease emerging risk and intensifying threats 2025. So we got three presenters, and then the first 30 minutes, it's a lecture that Bala Murali Suntram is providing. Then there are two case studies. So the 15 minutes each. So first case study by Anders, and then the second one is by Rachel Law. Then we have 24 minutes for the question and answer. So then you can usually, put it in your chat box, and then depends on the thing, then we can accommodate few questions and with the answers from the panel. Then we have a closing remark. So that with this kind introduction, I am starting that to introduce Dr. Bala Murali Sundram is a public health medicine specialist and senior lecturer in the Occupational and Environmental Health Unit, Department of Social and Preventive Medicine, Faculty of Medicine, University of Malaya. So he has led multiple national and international collaborations with agencies such as UN and University International Institute for Global Health, Malaysia, and then many agencies. So currently, he also active in the green health and sustainability initiatives at University Malaya, where he served as on the steering committee for the Green and Health Task Force. And also, he is a kind of a worked in Ministry of Health and also supporting for achieving carbon neutral health facilities by 2045, and reinforces the National Climate Change Policy 2.0, Malaysia's framework for low carbon climate resilient economy. So, so Dr. Bala, that the talk is today about exploring how climate change alters environmental conditions, expanding vector distribution to new areas, and the role of surveillance in monitoring and controlling disease spread. Dr. Bala, this over to you.
Okay. Thank you, Dr. Zurain, for a kind introduction. Okay. Okay. Hi, everyone. I'm Dr. Bala from the Social and Preventive Medicine Department, University of Malaya. I am a public health medicine specialist here, and it's been an honor to give a talk on climate change and vector-borne diseases, emerging risks, and intensifying threats. This is a very something close to my heart, coming from the background of public health and surveillance and environmental health. So let me start off. As you know, global health refers to a complex environmental change that occurs around the world. And it encompasses various issues, and including atmospheric composition and when we think about it, why vector-borne diseases are changing, we have to look at the broader scenarios of global change. So human activity has transformed the planet through urbanization, land use, deforestation, various other activities. So these interconnected drivers, as you can see, disrupt ecosystems and alter the natural balance between hosts, vectors, and pathogens. So I like this slide because it gives an overall snapshot of the impacts of climate change on human health, and it cuts across how it's got a direct effect on health. So we are seeing more frequent and severe heat waves, which increase the risk of heat exhaustion and heat stroke, particularly among vulnerable groups and elderly and outdoor workers. So these extreme heat events like the floods, storms, and droughts are causing a lot of deaths, injuries, and large-scale displacement. So you can see a multitude of effects as a result of climate change. And these can be immediate, long-term, and visible and often devastating. So climate change also has indirect health effects, which can also be dangerous, as we can see from here. Most importantly, from today's session, climate change, we have to understand that it reshapes the ecology of infectious diseases. Even a small rise in an average temperature can shorten, for instance, mosquito breeding cycles, while new rainfall patterns create more breeding grounds. So these indirect effects actually unfold when they unfold over time, they can have a long-lasting effect. So as we come to vectors per se, just a brief introduction, what are vectors? Vectors are living organisms that transport and inoculate causative agents or infectious disease. So anything that carries any infectious disease is called a vector. So, and invariably, this plays a very important role in public health and the spread of infectious disease. And most importantly, as we look into, there are many arthropods or vectors like sandflies, mosquitoes in particular. Their survival life cycle depends on the environmental condition, and indirectly, this itself has a direct effect on the human health. For example, and in other words, climate change per se has a role in this because, for example, warmer weather can accelerate mosquito development. It can actually reduce the development of the mosquito for a shorter intrinsic incubation period and causes you get more adult mosquitoes in a faster duration, and this can be infective and transmit diseases. So by understanding vectors as a living organism shaped by the environment, we can see why climate change has such a profound effect on its spread. So these are other arthropod or vector-borne diseases. We have sandflies, Lyme disease. So there's a myriad of non-mosquito origin vectors here. And this table is a nice table to look at vectors transmitting. What are the type of common vectors transmitting? This is a non-exhaustive table, but it actually depicts the major diseases and the related vectors which it transmits, right? Climate-sensitive vector-borne diseases, something very interesting and actually the forte of this talk. As we know, vector-borne diseases like parasites, viruses, and bacteria collectively cause more than 700,000 deaths every year. So some diseases especially are sensitive to climate change, and especially what you see here, most of them are sensitive to climate change. Mosquito-borne diseases like dengue, malaria, yellow fever, these Zika, Chikungunya are highly dependent on temperature changes and rainfall. Tick-borne diseases like Lyme disease also spread in a higher rate or in altitudes in a warmer climate. So we also see zoonotic spillovers like Nipah virus and Hantavirus, which become more prevalent in a climate stress condition, which pushes animals closer contact with humans. So it's very interesting, and this is a lovely table on climate-sensitive vector-borne diseases. Now, let's go to dengue, something very, very close to my heart because in Malaysia, dengue is always a perennial problem. I'm coming from Malaysia, and it's one of the most prevalent viral infection transmitted by the Aedes mosquito. And as you can see, the Aedes mosquito is a very, very highly domesticated, very intelligent breeder, highly anthropophagic, and it's a very smart mosquito where it's got the scripting breeding patterns, and it's highly adapted to our human population over time. So the challenges in actually eradicating or totally eliminating this, it's a huge challenge for us, and also in the spread of the dengue virus. So we can see transmission seasons in respect to climate change are getting longer, and dengue is appearing in newer regions, places where we never had, we started getting dengue infections with evolving serotypes. You can see that in the colored graph, that whenever we have a serotype change, you have a higher number of cases or deaths being reported. And usually when we look back in the Malaysian trend, or even worldwide, it's about every 5 years you get a serogenic shift in the serotypes. And this makes dengue a leading example of how climate change amplifies the disease transmission, and how Aedes or this particular mosquitoes actually acclimatize to the human population. Right? Coming to malaria, a brief note on malaria, one of the oldest diseases, infectious diseases we found. And as you know, malaria has shaped human health for centuries and remains a major killer of today until now, affecting almost 40% of the population. And WHO estimates that there are 350 to 500 million cases of malaria worldwide, in which 270 to 400 million are falciparum, the worst kind, and most are severe forms of malaria. So it's devastating, and we know that it's got a direct effect with climate change as well. As we see, the malaria problem, global problem, it has spread, and it's gotten much more significant in a way that there's a shift in the malaria transmission. Warmer temperatures actually cause the parasite incubation period to be shortened, so inside the mosquito, making them infectious faster. So this means there's more frequent biting, more efficient transmission, and seeing malaria spread into highland regions in which never occurred, of Africa, Asia, and South America, where it was once too cold, but because of warmer climate, there's already transmission of malaria. All right. And coming to yellow fever. Well, yellow fever, we thought it's something new. Some places of the world we wouldn't have heard of yellow fever, but you must remember it's the same vector. It's the Aedes aegypti as well. So Aedes aegypti transmits dengue, transmits yellow fever, transmits Zika, Chikungunya. So it's the same vector. So with world travel, international trade going on, and how the world is becoming borderless, mosquitoes can just jump onto the flight and land in another country within hours. So there's always a vector out there to transmit the diseases, right? So in terms of yellow fever per se, we can see that with the urbanization and climate change, the potential outbreak for yellow fever is actually quite prevalent now, and it's always there with the transboundary movement of travelers. Right? The one on the right, upper right one, is the yellow fever vaccination booklet. So anybody who's going into an endemic area of yellow fever, they are required to take the yellow fever vaccination. And now it's actually lifetime. It gives you a lifelong immunity. So you just have to take once in your lifetime if you are traveling to any endemic areas of yellow fever. Okay. Nipah virus. Well, Nipah is something close to Malaysia, because this virus was first isolated in Malaysia. There was an infection here. In the late 1990s, 98, 99, there was an outbreak of Nipah virus. And that was when it was initially an unknown disease. Then we realized that it's a new disease, and we named it Nipah, because Nipah was a valley or a small village in Malaysia. And that's where the Nipah virus occurred, in a pig farm. So, and recently there was an outbreak in Kerala, India as well, and they've linked it to bat habitats and deforestation and infected fruits, and the intermediate hosts like pigs and cows, they eat this infected fruits which are eaten by the bat, because the bat is already infected with the virus and it gets transmitted to the humans. So they can be a direct transmission, indirect transmission, or even directly to the intermediate host, right? Leptospirosis. Well, this is a condition or a disease where it's quite prevalent in many countries, in tropical countries, where as soon as the monsoon season or soon after a climatic effect as a result of floods or heavy storms, you can get a rise in leptospirosis cases. And because it's highly related to stagnant waters and high infected waters due to rat urine, most of it is caused by rat urine or rodent urine, or feces or droppings. And any other infected animal can also transmit, rodent infested animals can transmit leptospirosis. So it's usually quite prevalent in the cases during the monsoon seasons. And you can see in Malaysia, the cases usually are on the rise during the monsoon. And I remember there was a very heavy downpour in 2014, 2015. That's why you can see a lot of cases there, a lot of deaths. And over on the right hand, you can see the Thailand, and especially after the monsoon season, you can see a rise in cases. Other vector-borne diseases, let's not forget, it's just not through the mosquitoes. You have other types of vectors as well. And you have the CC flies, and what do you call, and ticks. Ticks are another common cause of vector-borne diseases. And usually these diseases are quite prevalent in certain countries, but it's highly adaptive, it's highly prevalent now, and you can see more occurrences due to climatic effects. And together they show that climate change affects not just one or two diseases, but an entire spectrum of infections across globally. Hantavirus, just a brief note, many of you might not have heard of this. It's actually from a rodent as well, and it's a zoonotic disease transmitted primarily through rodents, and particularly inhaling aerosol contaminates with rodent urine, droppings, or saliva. So unlike mosquito-borne infections, hantavirus is carried directly by small mammals. They look cute, they look small, they look harmless, but actually they carry a lot of viruses and infections in them. So next time when you see a rat, run away from it. Okay, climate plays a major role in shaping hantavirus outbreaks. After periods of heavy rainfall, yes, when you see a vegetation growth increase, and it actually boosts rodent population by providing more food and shelter. So as rodents increase in number, so does the likelihood of human contact with contaminated environments. So what's worrying is that climate change alters rainfall patterns, drives ecological imbalances, and you're likely to see more rodent population surges with them. And of course, increased hantavirus infection and other infections as well. This is a nice figure which maps different diseases according to your climate sensitivity on the. So you can see on the vertical axis, the climate sensitivity part, and on the x-axis, the health burden measured in DALYs. So on the far right, you see malaria, and so it has both a very high health burden and very high climate sensitivity, making it one of the most concerning diseases at the moment. And in the upper left corner, you can see Japanese encephalitis, the one circle, Rift Valley fever, West Nile yellow fever, Lyme. So all these diseases are highly climate sensitive as well, even if their current health burden is lower than malaria. But these are exactly the diseases that we have to look out for when there's a climatic effect. And this highlights that climate sensitive diseases are not necessarily the largest burden, but they're also the most likely to worsen under climate change. It's a nice table. And factors influencing emerging and emerging vector-borne. I think we can't run away in the public health side, the epidemiological triad, whether it's the agent, whether it's the host, the environment. So this is a very important, it's the bread and butter in public health, on how infectious diseases spread. So when you want to contain a disease, you have to target it in one of these parameters. So when you look at the drivers of vector-borne diseases, the pathogen itself plays a crucial role. Pathogens vary in their virulence, infectivity, and resistance patterns. So for example, some strains of dengue virus associated with more severe common outcomes. And like malaria, falciparum, it's a more severe ones actually develop resistance over time. And pathogens per se, they can adapt genetically due to climatic effects. So they became smarter. Mutations happen, and neurotransmissible vectors, mutations which are there, they can become more transmissible by new vectors, as seen in Chikungunya and certain other infectious disease. Coming to the host factors, as you can see, host populations, the second major factor shaping vector-borne disease dynamics. So the vulnerability depends on age, nutritional status, and previous susceptibility, prior exposure, and all that. Of course, vulnerable populations, especially children and elderly. Another issue is co-infection. So individuals living in regions with high HIV prevalence may be more susceptible for severe malaria or tuberculosis due to poor nutrition and climate-related food shortages. So this can also aggravate it. And let's not forget the environmental effect. So in this, we have our physical structures, the climatic effect, which are interactive with the other agent and host factors. And you have all the other issues like sanitation, climate, housing, and urbanization, which can also accelerate this. And these are certain examples that, we must also not forget, yes, biodiversity causes and other environmental factors which are being rapidly transformed by global change and making them an important driver in the spread of vector-borne diseases. This is a direct climate effect on disease vectors. So you can see when there's a change in temperature, global wind patterns, precipitation patterns, and changes in relative humidity, there's always a change in way the vectors behave, and indirectly it leads to a higher spread of disease to some extent. And like for example, when there's a change in the global wind patterns, it can alter the migration of certain insects, certain vectors, and it can actually accelerate the transmission of zoonotic or vector-borne. Effects of temperature. Let's go just for temperature starting off. And temperature, as you know, is one of the most critical climate drivers of vector-borne disease. As the temperatures become warmer, the climate becomes warmer, mosquitoes and ticks develop faster, and reproduce more quickly and feed more often. So it's another way that it becomes an accelerator for the spread of vector-borne diseases. And you can see that's how they adapt, the pathogens. And warmer environment shortens the time the parasites need to incubate inside the mosquito, meaning more vectors become infectious in less time. Okay. Comes to and other vector-borne diseases like ticks and also replicate similar patterns and similar way on how they develop and reproduce and then spread the infections. Rainfall, another important factor due to climate change, where there's increased precipitation, creates more potential breeding sites for mosquito. And when the vegetation is dense and after rainfall, this provides more shelter, resting grounds for vector, and this itself can actually be more conducive for breeding. And heavy rainfall, larval development creates increased vector capacity and outbreaks. So you get a lot of clusters and outbreaks happening, and this itself accelerates transmission of diseases. Soil condition, something very important and something we always miss out. Soil moisture is a factor in the development stages of ticks. So this is another important, often overlooked role in disease transmission. So the soil moisture composition actually play a vital role in the transmission of certain hosts, and indirectly they can actually lead to the spread of certain diseases, right? Ocean temperature, sea level, and acidity. Well, rising sea levels, very important, and you can see the sea level rise going up with the climatic effects, melting of glaciers. So you can see that there's always some brackish water where what seawater it goes back and infiltrates into the water bodies inland and increases saltwater intrusion in freshwater sources. So there will be some sort of vector adaptation, and certain vectors actually can adapt and tolerate to brackish waters. That's why we are getting outbreaks of malaria in some marsh areas, and due to certain species like Anopheles and Ricus, and that's how they adapt. And these are the pathogens, actually, they do the adaptation, and due to the environmental stress which they go through, they go into adaptation, viral mutations, and this can actually enhance the transmission efficiency. So you can see this is an example where this is in the Andaman Nicobar Island, where there's some, due to sea level rise and brackish waters, you can actually, there were incidents of malaria transmission, and we realized that was these vectors actually adapted, and they became more saline tolerant mosquito larvae, and they could actually adapt to the salt water and breed and actually transmit the disease. During the Asian tsunami of December 2004, there was a huge tsunami at that point in time in parts of Asia, and following that, there was a widespread malaria infection. So that's how we can see the pathogens are becoming much smarter and adapting to climate change. Drivers of global change affecting dynamics of vector-borne disease, as you can see, we have atmospheric composition, one of the very important factors of global change in atmospheric composition, greenhouse gases, carbon dioxide, methane, and nitrous oxide. So these are another good, important factors that lead to vector-borne diseases. Climate change, I explained just now, temperature, precipitation, which warmer temperatures speed up mosquito and parasite development, while higher humidity improves vector survival. So this is another important factor. Urbanization. We're massively going into urbanization process, and it's a very critical driver in the transmission of vector-borne diseases because it's unplanned, it's rapid, and many cities are low, middle income, and more countries face challenges with water supply, drainage, waste management. So these are the core of leading to transmission of vector-borne disease. And another thing, of course, human trade, human movement, global use, industrial agriculture, biodiversity, land use. These are also shaping the way the disease dynamics flow. And when it comes to deforestation for agriculture, logging, and settlements, it alters the habitats and disrupts long-standing relationships between the vector, hosts, and pathogens. Globalization, I mentioned, seamless traveling. So the world has become borderless. You can get infected in one country, within hours, you can spread the disease to another country. We saw that in COVID, and vectors can easily transmit that as well. Right? This is a nice table looking into all the global change drivers, which are the climatic effects of temperature, rainfall, urbanization, and all the others, and how actually it affects on the vector, pathogen, and host environments, and what it leads to. So it's very summarized nicely here. And just a brief note on re-emerging diseases. You can see that we're getting this close. It's closely tied to climate change, and although 50 to 60% are zoonotic, but when you look at newly emerging diseases, the 75% new diseases come from animals. So on an average, a new human infectious disease emerges almost every month or every 6 to 7 months. And climate change accelerates this process by altering habitats, changing vector distributions, and stressing ecosystems. So the result is more frequent opportunities for pathogens to jump from animals to animals, and then finally affecting the humans. Adaptation. So how do we come to this? Just a brief note on mitigation and adaptation. To respond effectively, we need a dual strategy, which is mitigation and adaptation. Mitigation focuses on addressing the root cause by reducing greenhouse gas emissions, shifting to renewable energy, maybe pursuing sustainable use of land. And adaptation, on the other hand, prepares health systems and communities to live with the changes which are already happening. So these are important factors when it comes to climate change. And we can use this as a mantra to actually counter vector-borne disease transmission as well. One Health approach, very important in terms of zoonotic vector-borne diseases. It's a framework that emphasizes interconnectedness of human, animal, and environmental health. So the principle is simple but powerful. And if we ignore the health of ecosystems, I know we cannot safeguard human health. It's very important. And this whole concept of One Health is an important framework. Strategies to combat vector-borne diseases. various strategies. There's a lot of strategies out there. In Malaysia, we started this Wolbachia inoculation. So it's to help to control mosquito population. Wolbachia is a type of virus. It's a bacteria actually, and it's injected into the mosquitoes, and any Wolbachia infected mosquitoes can't breed. So indirectly, it lowers the population of Aedes aegypti. So this is more like a biological control and genetically modified. But we have other methods, that genetic modified methods, and I think it's here with genetic modifications to combat mosquito-borne diseases. And we also have developed decent techniques for combating mosquitoes by implementing mental management, insecticides, sterile insect techniques, and other pathogen blocking bacteria. And in public health, one of the most important factors is developing early warning systems and protection measures. So it's very, very important that it's an important component in public health, where we need to new innovations to combat spread of disease. The framework for designing adaptation options for the management of vector-borne diseases, well, it's important because there can be various methods. So we can look into the vector control and targeting the pathogens and in one way. And of course, the successful control vector-borne disease depends on capacity of community ownership, surveillance, and various other public health delivery methods. So adaptive response can be in the form of legislation, so administrative actions and monitoring, case detection, surveillance, diagnosis, and immediate reporting, and notification, climate forecasting, computer modeling, and early warning systems. Behavioral change, important, so socially, so the community is involved. Personal behavioral responses is very, very important, especially to combat infectious disease spread, avoidance of exposure to vectors, by staying indoors. Mosquito breeding time is usually in the dawn and dusk, so we need to be cautious about that, wear protective clothing. These are simple behavioral techniques. And of course, engineering methods, we have all this insect repellents, bed nets, pesticides, repellents. So these are all chemical and biological way of controlling spread of the disease. IPCC, just a brief note, it's an intergovernment panel on climate change, and we've actually were involved. I was actually involved in this as well, during the third national communication and second biennial update report. So we stress on this report, we stress on our adaptation, mitigation strategies, and vector-borne control, and it does give a framework for us to share with the other countries, right? In conclusion, climate change is one of the greatest challenges to modern human civilization, as you can see, and we can see there's a shift. Even a small shift makes a difference, and a small shift can not only dynamically it can cause a higher difference in weather and climate, but it's more than that, and it directly affects our health and the distribution and behavior of parasites in their hosts and vectors. Climate change will cause increase in disease incidence. I spoke about re-emergent and diseases, and it's essential to develop a portfolio of strategies which include adaptation, mitigation, and other technological developments in combating climate change. Thank you. With that, I hand my talk over to you, Dr. Vajan.
Thank you so much for this elaborating your experience with this, particularly the context of Malaysia, handling of vector-borne disease, and then related to the climate and how it has kind of an impact on that. So now we are moving to the second phase of that this program of climate change and vector-borne diseases, emerging risk and intensifying threats. The first case study is going to be the next 15 minutes by Katie Anders on examining the use of technology and targeted interventions such as Wolbachia and vaccination to control vector populations and prevent disease outbreaks. So, Katie Anders is an associate professor and National Health and Medical Research Council Australia Research Fellow in the School of Public Health and Preventive Medicine at Monash University, Australia, and a member of the Monash Health Climate Initiative. She leads applied public health research and implementation research to inform the targeting and scale of effective strategies for controlling dengue and other vector-borne diseases, and has research interest in the interactions between local and macro drivers of arboviral disease epidemiology and intervention effectiveness. He has extensive international experience in arbovirus disease epidemiology and the design and the implementation of field trials. So, including 10 years at the Monash-based World Mosquito Program, establishing the evidence base for Wolbachia-based dengue control. So, she is the most appropriate person to share some of her experience today. Welcome, Katie Anders, and please take it up.
Thanks very much for that introduction, and thanks to the previous speaker for a really comprehensive overview of all of the pathways by which climate can impact on vector-borne diseases, including dengue, which is going to be the focus of my talk today, and specifically presenting a case study on an innovative intervention for prevention and control of dengue and other viral diseases transmitted by Aedes aegypti mosquitoes, which I've been working on for the past 10 years, which is the Wolbachia intervention that was mentioned briefly in the previous presentation. And just a little bit of background, which I think many of you will be aware of, the fact that recurrent dengue outbreaks have occurred throughout tropical and subtropical regions for decades, but that the size and the frequency of dengue outbreaks and the number of countries affected has really increased dramatically over the past 2 decades, with a record 6 million cases reported to WHO in 2019 and 2023. And then last year, really seeing a surge in dengue transmission throughout many Asian and Latin American countries, with cases exceeding 14 million reported cases, more than 10,000 deaths, and concurrent Chikungunya and Zika outbreaks occurring in many countries as well. So this is really the challenge, the unmet public health need that we're facing, with climate change and rapid urbanisation really being key drivers of this resurgence. And half the world's population currently estimated to live in areas at risk of local dengue transmission, and that projected to increase over coming decades due to warming temperatures combined with population growth in tropical cities and expansion of the range of the Aedes aegypti and Aedes albopictus mosquitoes. So I just give that as the sort of scene setting, together with just a brief bit of background on what the sort of status quo is around dengue vector control. As mentioned, the primary vector is Aedes aegypti, which is a domesticated mosquito that's really well adapted to live in and around human habitation, breeding in small artificial water containers, biting humans, being inside and biting during the day. And so the mainstays really of dengue vector control in endemic countries have been mosquito surveillance, identify and remove or reduce the breeding sites for these mosquitoes through public health messaging and cleanup campaigns, shown on the top here, tires, any sort of trash around properties, or even these really small amounts of water at the bottom of pot plants, etc., ideal breeding grounds for larvae, Aedes aegypti larvae in and around the home. And then the other sort of arm of conventional control has been chemical control with the use of larvicides targeting immatures, so larvicides into water sources, and insecticides spraying to try and kill adult mosquitoes. But these approaches have really been limited in their effectiveness at scale due to high levels of insecticide resistance and the difficulty of sustaining these activities and scaling these activities in large tropical settings. And with that fogging shown on the right-hand picture there, really not reaching a lot of those adult mosquitoes that are preferentially indoors. So I've just included here a couple of papers that are quite old now, 10 years ago, really highlighting some of these limitations in conventional vector control, which is really led to a recognition of the urgent need for the development of innovative interventions for prevention and control of dengue, and the integration of innovative approaches into public health programs and their scaling within the context of integrated vector control. So this is one WHO document that highlights in its pillars of action this WHO Global Vector Control Response Strategy, highlighting the need for innovation and the monitoring and evaluation of interventions to enable their scale up and integration into dengue control programs. And another WHO sort of strategic document, strategic guidance from last year, the Global Arbovirus Initiative, which goes through 6 different pillars that need to be strengthened for effective arbovirus control, one of which again highlights the need for enhancing innovation and new approaches and generation of evidence for effectiveness and how those can be optimally scaled and implemented alongside other tools, both conventional tools, but also other emerging interventions like vaccines that are becoming available for dengue and chikungunya in recent years. So I've showing in this figure, the tools that are available for dengue control, as I said, mosquito surveillance and control, diagnostics, therapeutics, and clinical management. There's no antiviral for dengue or specific therapeutic, but a lot of effort going into the development of candidate drugs and therapies. We'll hear in the next talk about innovations in outbreak early warning and prediction and disease surveillance, obviously being an ongoing mainstay of dengue control. But the 2 new interventions that have become available in recent years are licensed vaccines for dengue and chikungunya, so individual level interventions which give individual level protection, and Wolbachia-based control, which is delivered at the level of communities or city level for population level protection. So I'll be focusing on the Wolbachia-based control for most of this talk. And what this is a mosquito-based intervention. Sorry, yeah. Yep, that's showing properly, which involves the release of mosquitoes that carry a bacterium called Wolbachia. This is a naturally occurring bacterium that lives inside the cells of around half of all insect species, including many other mosquito species, but not Aedes aegypti naturally. And the way so Wolbachia is inherited maternally in all insect species in which it exists. It's transmitted vertically from the parent to the offspring via the insect's eggs. And what makes Wolbachia useful as a public health tool was the finding that when it was purified from another insect species, so that could be the Drosophila fruit fly in the case of the W. Mel strain that the World Mosquito Program uses, or W. alb strain from Albopictus mosquitoes, Wolbachia was purified, micro-injected into the eggs of Aedes aegypti, which is shown in the middle picture there, and those Aedes aegypti were reared up into a colony of maternally inherited Wolbachia that's stable in that Aedes aegypti population in the insectary, and then how that is operationalised as a public health intervention is that when Wolbachia-infected Aedes aegypti are released into a city where the dark blue is just wild Aedes aegypti mosquitoes, Wolbachia-infected Aedes aegypti in light blue are released over a period of several months, either as eggs in capsules, shown in the top two pictures there, in a container with some water and some food source, and they emerge and they fly out into the community, or as adults, as shown in the right-hand two pictures there, with high-density releases every 50 metres or so for a number of weeks. Those Wolbachia mosquitoes mate with the wild Aedes aegypti, pass Wolbachia through the population, and after a period of time, due to the maternal inheritance and a reproductive advantage that the Wolbachia mosquitoes have, those Wolbachia mosquitoes replace the wild type population, and Wolbachia self-sustains in the Aedes aegypti population going forward. And so after releases stop, Wolbachia is stable. So this is counter to the approaches that aim to reduce the mosquito population. In this case, it's replacing the mosquito population, and the effect that Wolbachia has on the Aedes aegypti is to, within the mosquito cells, compete with the virus for resources such that Wolbachia carrying Aedes aegypti have a significantly reduced ability to transmit dengue and other viruses, including Zika and Chikungunya. So it's replacing the population with mosquitoes with far lower competence for transmitting these viruses. And so what I'd like to highlight in my remaining slides is really in a very short space of time to summarise the way in which over the last 15 years this has moved as an innovative technology from the lab to the field to now scale up in multiple countries, and the types of evidence that have been needed to be generated, and importantly the cross-disciplinary capabilities and competencies that have been critical in taking Wolbachia from the lab to the field to scale up in multiple countries in the space of 15 years. So this is from a WHO guidance document on trial design for developing and trialling new vector control interventions from phase 1 through to phase 3, phase 4, which will be familiar to many people. But what I've added on the right here are the numerous other domains in which evidence needs to be generated in order to transition from research to programmatic uptake. And what should be apparent from this is that it requires, in addition to the core sort of entomology, laboratory, epidemiology expertise, also multidisciplinary expertise in economics, regulatory affairs, public health policy, financing, and a broad range of partnerships in order to achieve that. So I'm going to skip through very briefly. I won't go into the details here due to a lack of time, but the stages in which this evidence generation was conducted for Wolbachia was initial proof of concept field trials in Northern Australia, where it was intended to show that Wolbachia could be established in the Aedes aegypti population. So what this graph shows is the proportion of Aedes aegypti in the field in these two areas of Cairns in Northern Australia, which when they're tested in the lab, they contain Wolbachia. So the green shading is when releases were conducted in early 2011, and all the other data points are from long-term monitoring after releases have stopped, showing that Wolbachia stays established at a very high level in the mosquito population over time. The pivotal efficacy evidence for Wolbachia was generated in a cluster randomised trial in Yogyakarta, Indonesia, and I've given the reference there. I won't go through the trial design here, but these graphs show the rapid establishment of Wolbachia in the local Aedes aegypti population in the 12 clusters shown in dark blue on the map, and the top graph in the middle where Wolbachia was released and was established stably over time. There were 12 untreated clusters, and through prospective enrolment of febrile patients in primary care clinics and testing in a reference laboratory, we were able to show that there was a 77% lower incidence of virologically confirmed dengue in residents of the Wolbachia-treated areas versus untreated areas, and that was consistent across all 4 dengue virus serotypes. So a key consideration for a novel biological intervention is, of course, safety and the potential for adverse effects, either for humans or the environment. So I've included here a number of the scientific studies that have been published looking at different aspects of potential concerns around Wolbachia. Ecological, or so these studies show that there's no horizontal transfer of Wolbachia to other species or not to predators. Humans bitten by mosquitoes carrying Wolbachia don't make antibodies to Wolbachia. Wolbachia cannot infect vertebrate hosts like humans. It's only able to infect insects. And it exists in the environment in many different insect species, including biting insects naturally, which gives us a lot of confidence that it's safe for humans and the environment. And there were independent risk assessments done prior to the first releases in Australia by the National Scientific Organisation in Australia, as well as in Indonesia and Vietnam, all of which concluded that the risk of harm from Wolbachia releases over a 30-year horizon was negligible, and there have been no adverse events attributable to Wolbachia releases reported from any sites. Very quickly, in the last few minutes, since the first releases of Wolbachia in northern Queensland, a core component of all the release programs has really been community and stakeholder engagement for a several-month period leading up to the first releases, because this is obviously sort of a counterintuitive intervention, releasing mosquitoes to combat a mosquito-borne disease. And so I've included here what's called the public acceptance model, which has been implemented, locally adapted and implemented in each of the countries where Mosquito Program has partnered with governments and communities to deploy Wolbachia mosquitoes, really focused on raising awareness, providing pathways for communities to access information, raise concerns through community reference groups and other means, and measuring the community sentiment and awareness and acceptance of the intervention. And the nature and strength of the partnerships in different countries, I think, has really been a key enabler in both the successful outcomes of initial projects, but really importantly in having then a pathway for expansion of beyond an initial pilot project and demonstration project by having ministries of health engaged early on, as well as civil society and other academic partners. The last thing I'd like to show here is about scalability and reproducibility. So it's one thing to show effectiveness or efficacy under trial conditions, but from several locations where Wolbachia has been deployed across whole cities, so including Yogyakarta in Indonesia on the top graph on the left here, and a city of 500,000 people in Brazil called Niterói. What we've seen is that once Wolbachia is well established in the local mosquito population, it self-sustains over time, and you see this disruption of the historic seasonal epidemics that are typical of dengue epidemiology at baseline in these countries, to then with the blue shaded area showing that phased rollout of Wolbachia across whole cities. You don't see flatlining, but this is notified dengue cases here from the disease surveillance data. You really see this sustained interruption of those previous epidemic cycles. But I do want to highlight that in a few release sites, establishment of Wolbachia in the local mosquito population has been more variable. And one of the key drivers of this is understood to be climatic conditions, so very high temperatures having an impact in the lab and in some field sites, like we're seeing in central Vietnam on the top graph here, where Wolbachia prevalence in the mosquito population temporarily drops during the hot season and recovers and drops again and recovers. But in one site on the right with the map shown here, it's hard to see, but the green pie charts on the bottom show that Wolbachia has stayed well established, where the white pie charts on the top showed dropout of Wolbachia, which is thought to be due to extreme heat events. And in Rio de Janeiro in Brazil, also 3 neighbourhoods where Wolbachia was never well established and then dropped out, potentially due to ecological factors or the use of insecticides in that setting, combined with a fitness cost of Wolbachia on those mosquitoes. So this is my last slide. I just want to highlight finally the fact that the policy and regulatory pathways for a novel intervention like Wolbachia don't exist. So these have, it's not like vaccines and insecticides and other technologies for which there's regulatory and health technology assessment pathways. For an innovative tool like Wolbachia, the World Health Organization Vector Control Advisory Group has responsibility for evaluating the evidence for public health value. They, on the basis of the dossier of evidence submitted, concluded there was evidence of public health value and have recommended WHO to develop a policy guidance for Wolbachia, which is still forthcoming. But in countries where the evidence base has, been established over the past 10 years, ministries of health in Brazil and Indonesia are moving forward with integrating Wolbachia into national arbovirus control policies and programs. But what is key here really is the need for locally adapted operational models that enable scaling, and that's context specific and requires evidence generation within the context of that country and what is needed for informing integration into dengue control programs alongside other strategies like vaccination as well. So I'll stop there and happy to take any questions at the end of the session. Thank you.
Yeah, it is a very wonderful, excellent lecture. And now also you have really have gone through all these, how this evidence is created, and then making it to know that community engagement, and then later on that how these things are working and the policy at the national level and also international, and how things are progressing to adapt this kind of a thing. So it's a very good case scenario that for all of our participants and here to how that evidences could be developed and translated into real practice, and also it's a thing then understanding what is happening at that introducing that Wolbachia into the control of arbovirus and the dengue prevention. So thank you so much, and we will take it up a couple of questions after the second case study and before ending the sessions. So now we are moving to this second case presentation is by Rachel Lowe. That she's going to discuss with us about early warning systems for vector-borne disease. Professor Rachel Lowe is an ICRA research professor and global health resilience group leader at the Barcelona Supercomputing Center. The EHR group, the core develops policy-relevant methodological solutions to enhance surveillance, preparedness, and response to global health challenges with focus on climate sensitivity, infectious diseases. Rachel coordinates two Wellcome Trust digital technology, climate and health projects, Harmonize and IDX teams, which aim to provide robust data and modeling tools to build local resilience against emerging infectious disease threats. Rachel is a member of World Meteorological Organization, World Weather Research Program, WWRP, and the Lancet Commission for strengthening the use of epidemiological modeling of emerging and pandemic infectious disease. Rachel was nominated as a lead author of IPCC's 7th Assessment Report, chapter on health and well-being. So, Rachel, we are very happy to listen from you, the early warning systems for vector-borne diseases.
Thanks so much for the introduction, and thanks for having me. So it's a pleasure to share some of our research on co-creating early warning systems for vector-borne diseases. We've been investigating how we can best make use of climatic and environmental information to try and predict disease risk in advance. And we know that climate can affect infectious diseases in several different ways. We recently published a framework outlining ways that we can identify the climate sensitivity of an infectious disease, thinking about the way the climate can impact both the seasonality, the geographical distribution of a disease, and the interannual variation in incidence. And we know that anthropogenic climate change can lead to a shift in the seasonality. It can change the timing of onset or the duration of the transmission season. It can shift the geographical range limits, so we may see an expansion or a contraction, or we may see increased risk at higher altitudes, for example. And it can also increase the intensity and frequency of extreme events, which can in turn impact the severity of outbreaks. So we've been looking at how we can incorporate climate information into decision support tools across a range of timescales. So we look at developing indicators to track historical changes in climate-sensitive infectious disease risk given observations. Then we can look at short-term predictions using weather forecasts, or we can look at sub-seasonal to seasonal indicators, which can often feed into early warning systems that can help with planning from, say, 1 to 6 months in advance. And we can also look at climate change projections, looking at different scenarios depending on the amount of CO2 emissions that are pumped into the atmosphere and the kind of policy and mitigation adaptation measures that may be taken by governments to be able to better motivate and implement mitigation adaptation plans. So a group of us a couple of years ago published a chapter in this book on planetary health approaches to understand and control vector-borne diseases. And we lay out our experiences in developing early warning systems for vector-borne diseases with a focus on engagement methods and implementation, and how all this fits together to eventually lead to some kind of tool that could be sustainably implemented in practice. And a lot of this is very much dependent on strong partnerships to be able to develop tools that can be locally owned and sustained, and that can provide routine information to allow ministries of health or disease control programs to be able to have some advance warning of the potential increased risk of an outbreak in advance. This is the experience of a partnership we developed in the Caribbean, particularly with the Caribbean Institute for Meteorology and Hydrology and the Caribbean Public Health Agency, together with national agencies in Barbados, including the Meteorological Services and the Ministry of Health and Wellness. And this is a partnership that's built over the years, which thanks to several different funding sources, so we've had to sort of string together these little pots of money, but that did eventually allow us to build together a strong partnership, which has led to some initial implementations. And also working with the interdisciplinary team of researchers across different institutes and countries, as well as the key stakeholders. So this has led to both methodological and modelling publications, as well as different kinds of publications outlining our co-learning experience of developing these early warning systems and an implementation framework to implement a climate services for health, particularly in the Caribbean setting. And this work was also included as a case study in the 2023 State of the Climate Services for Health report, published by the World Meteorological Organization. So after several years of research identifying these delayed and nonlinear impacts between different extreme events and how we can best incorporate them into the services which are operated by the CIMH and the meteorological services, we've developed a scheme which relies on the interactions between long and short lag indicators that can tell us about the potential drought or exceptionally wet conditions, together with information about the temperature. And this can lead to different levels of dengue risk outbreaks. So, for example, if we want to issue a forecast 3 months in advance, we can observe the standardized precipitation index in the previous month and combine this with the current temperature conditions at the time of forecast, and combine this with the forecast of the SPEI, the drought indicator for the following month. And if we're to see a scenario of dry, then hot, then wet conditions, this would lead to an increased risk of dengue. Whereas if we were to observe dry and cool conditions and the forecast were for conditions to continue to be dry, then the model would issue a lower risk of dengue. So this kind of information is being implemented on a routine basis and was used last year operationally ahead of the 2024 Cricket World Cup tournament that took place across the Caribbean. We've also been using these climate-driven modelling approaches together with the National Environment Agency in Singapore to help understand the potential effectiveness of their recent Wolbachia program, which was ramped up in 2023. So to be able to do this, we formulated a dengue prediction model that incorporated both climate information with information on changing serotypes in Singapore. And this model was then used to estimate the potential number of dengue cases that would have been experienced in 2023. And because this was fitted to data before the Wolbachia intervention was implemented, we could then use this as a proxy to the potential number of cases that may have been averted according to this intervention. And we found this to be approximately 28% reduction in cases. So this paper should be hopefully published soon. So I hope to be able to share it with you then. We've also been working on more efficient ways to be able to harmonize multi-source, multi-scale data to better inform vector-borne disease risk management strategies. And in this annual review of entomology paper, we laid out a data harmonization process framework to think about the different steps that are required to bring together all these different types of data, which may include gridded data sets, it may be point data from weather stations or sentinel sites, it may be travel data, at different scales from global to local. So to be able to do this, we need to first understand the question for which we need this data harmonized. We need to design any new data collection. We need to audit any existing data. We need to set up data sharing agreements, which can be a very lengthy process. We need to quality assure the data, post-process, and bring everything together on a common space-time resolution. We need to document and share the data and code and make sure that's properly archived. We need to test and evaluate it with users, and we need to be continually disseminating and updating the findings and making sure that we can have robust and reliable data sets that can then be fed into these kind of early warning and disincidence support systems. And of course, there are many limitations to the kind of open source global products that are available for download. For example, if we're thinking about small islands, sometimes these gridded products are simply not fit for purpose, depending on the spatial scale. On the left here, we can see Dominican Republic, which is a relatively large island. And here we can see the ERA 5 land reanalysis product. This is the precipitation field mapped across the island, and this has a spatial resolution of approximately 9 kilometers squared. And here we can pick up some of the orography on the island. But if we're looking at the very small islands like Barbados, Grenada, and St. Lucia, then we have very few grid squares providing information. And in these settings, it's more preferable to be using the local weather station data to inform these kind of models. And a few years ago, we conducted an exercise to test the importance of the choice of the climate product within these kind of impact-based models. So with partners in Ecuador, we formulated a dengue and a malaria model in southern coastal Ecuador. And we found that depending on the temperature product we use, we could find quite different answers to the impact of temperature on dengue. So for a 1 degree increase in temperature, using temperature data from a weather station located in the city where the dengue cases were reported, we could estimate a 68% increase in the relative risk of dengue. Whereas if we were to use one of the gridded products, for example, the CREW product, then only a 40% increase in dengue risk would have been estimated. And in the case of malaria, we found that the precipitation data from the weather station gave the opposite answer to several of the products. So this is really a word of warning that these global products can't necessarily be taken off the shelf and plugged into a local level decision support tool. This is an example of some work we're doing as part of our Harmonise project, which is funded by the Wellcome Trust, where we're looking to harmonise different types of data and downscale course resolution data to the local level, taking into consideration different kinds of altitudinal or urban to rural gradients. This is one of our hotspot sites in Colombia. Here we can see in orange, this is the Cajamarca, which is within the state of Tolima. And in this area, we have installed 3 weather stations along an altitudinal gradient, and these complement the already existing weather stations in the region, which are marked by the red symbols. And we are then going to be, we're developing an R package which can allow us to understand how well the gridded reanalysis product, which is the blue grid, can represent the temperatures across this altitudinal gradient, and then how well can the seasonal climate forecasts, which have a much coarser resolution of around 100 kilometers square, that's the black grid, how can those forecasts be downscaled to the finer scale grid to then eventually provide some forecasts that could lead into some kind of disease prediction system. And a lot of these data collection and installation projects have been developed together with local communities and partners. In the study sites, these are some images from a field trip that we conducted in the Colombian Andes a couple of years ago. We can see here on the left, these are some this sort of illegal fly tip that was identified together with the community and could be observed using drones that were that were issued by the research team there. And here on the right, we can see one of the weather stations that we installed within a ecological reserve, and this station will continue to belong to the reserve after the lifetime of the project. So within the Harmonise, we're developing a toolkit of both R and Python packages that will allow users to be able to bring these different data types to a common space-time scale and then harmonize them together to develop an analysis-ready data set that could eventually be used for the kind of modeling examples that I will show you next. So we have a package called CLIM for Health and Data for Health, and partners across our consortium are developing Socio for Health, Land for Health, and some more advanced packages to be able to work with data cubes and also drone imagery. So once we have our harmonized data and we have our calibrated climate forecasts, we can then develop a statistical framework to understand the different links between these climatic, environmental, or socioeconomic indicators and the disease of interest, and then use this model to make some forward-looking predictions. And we can extend our lead time into the future by using forecasts of the climate variables. So we have some advanced warning of changing climatic suitability for these diseases. So here you can see the suite of our tools that we're developing all together within the Global Health Resilience Group. We're calling these the GHR tools. So first of all, we have our Data for Health and CLIM for Health packages, which can help bring the data to the space-time resolution you need. And then we have 3R packages that can allow users to explore, model, and predict the data. And our target users are based in with our partners in Barbados to bring these tools into an operational system, and with partners in Brazil to be able to extend the lead time of the existing early warning systems there. So these are just some examples of how the tools are being used. This is the dengue early warning that was issued ahead of the 2024 Cricket World Cup for the Barbados Ministry of Health and Wellness. And this model framework issued a probability of an outbreak of 95% ahead of the games. And we've also been developing different kinds of visualization techniques to try and more easily communicate these kind of risk levels to the users. And this allowed the disease control teams to ramp up their existing efforts, which had already been enhanced due to the event itself and many spectators from around the world. And this additional forecast allowed extra control measures to be taken, particularly around the stadiums. We've also been adapting our tools to be able to take part in the InfoDengue forecast sprints. So last year, we entered as one of several teams trying to reproduce the evolution of the dengue season in previous years to make an outlook for 2025. We can see on the left, the Global Health Resilience Model is marked in orange, and it quite closely matches the observed dengue incidence. This is for the state of the Amazonas state in Brazil. This is in the validation set. And then this year, we also took part, and we could implement our tools and the new methodologies that we've been developing. And overall, our model was one of the top scoring models, which is ranked in terms of the number of health regions and states for which the model correctly could predict the number of outbreaks and how well it did in the different regions. So this is quite promising for the potential utility of this model for supporting ministries of health in making decisions by incorporating climate information. And we're bringing all these tools together into an interactive platform. We've been developing this over the years with many partners in Latin America and the Caribbean region as part of our Wellcome Trust funded projects. So we initially put this together in response to the 2023 El Nino event, where the warming of the sea surface temperatures in the Pacific led to anomalous climate conditions in the west coast of South America, in northern Argentina. We tend to see flooding events in South Brazil and northern Argentina, exceptionally warm events on the west coast, and very dry conditions in the Amazon. So we're trying to bring together forecasts of these conditions to be able to make disease risk estimates and provide these to our partners as the El Nino event was emerging. So this is a platform that we're developing and we're hoping to improve and implement in other places over the course of our upcoming projects. And as well as the technological tools, we've also been putting together different kinds of frameworks. Here is some guiding questions for early warning system implementation. So being able to think about which different partners should be involved in which capacity, who is in charge of maintaining and funding any early warning system in the long term, what kind of outputs are needed. Should this be a PDF emailed out each month, or should it be an interactive dashboard? Who is responsible for monitoring and evaluation? And how can we communicate the return of investment to the public? And as well as the framework, we've also been gathering and conducting capacity strengthening and knowledge exchange activities. These are some examples from in the Caribbean and also in Asia, where we've been working with different kinds of partners, from medical officers to entomologists to climate scientists and academics, to work together to show how these tools can be used and the kinds of data and resources that need to be brought together to make this happen. So with that, I'd like to thank you all for your attention and thank my team and all our collaborators and our funders, and I'd be happy to take any questions in the Q&A. Thank you.
Thank you so much, Rashid, elaborating and then how the tools are developed and implemented, and how you are trying to expand. Then, because now we have a question and answer session, so that we don't have, because of this time, we have another 8 more minutes. So to all, I'm proposing this question to all. Given the growing health impacts of global climate change, what key strategies should be taught to public health students, drawing from your experience, to help them prevent, manage, and adapt to climate-related air pollution within public health system?
Maybe thank you, Dr. Rajendran. Maybe I can answer that in a public health perspective, being a practitioner in the field. And I think one of the most important message here is community participation when it comes to the risk of disease spread, because we're talking of vector-borne diseases and the effect of course, the climate drivers are there, but we need to get a message to the local community and need to empower them. That was a very important aspect in our campaigns and designing campaigns and solutions in a local community. So more of a community-directed intervention is very important in transmission of diseases in any health promotion activity. It's a challenge, yes, but we need to give empowerment rather than fear, and we need to get the local communities, the religious leaders. These community played an important role in passing out our message in combating these issues. And we realize that we are talking about health, we are talking about vector dynamics here and climate change, but it's also a social issue, and a community-driven approach is very, very important. Besides, of course, the public health approaches in terms of vector control, in terms of biological vector control, chemical control, and other aspects of it.
Okay, thank you. And in this context, I'm proposing this question to Katie. In the context of climate change driven expansion of vector-borne diseases, many interventions such as predictive modeling, genetic modification of vector or novel vector control technologies show promise in pilot studies. So how can we critically evaluate their scalability, reproducibility across diverse ecological settings and local feasibility in low resource contexts without compromising cultural acceptance, equity, and sustainability? It's a very long question. Is it? Are you able
I mean,
to?
it's a great question, and this is the challenge, right? I mean, you can't do trials in every setting, but equally there needs to be locally relevant data to understand variability in both the acceptability, the feasibility, and the effectiveness of these different interventions in different settings. That goes for Wolbachia, but also what Rachel's talked about with how these early warning tools and predictive models will work in different settings and be used within public health programs in different settings is going to be contextual. So I think it really comes down to engaging with, like, you need local champions, you need to have those partnerships early on with people who understand the need, understand the tool and the technology, and are the ones who within their own context can advocate for the role of that new tool within that setting and generate the local evidence and speak to the local evidence within their own context. It takes time, but I think it's really about having that local ownership and understanding of those innovative tools to be able to generate the evidence and to scale up.
Thank you, Katie. This is to Rachel. Rachel, then what are the possible ways we would be able to implement early warning system for the vector-borne disease without overwhelming the already fragile health service system of the low middle-income countries?
Thanks for the question. Yeah, that's a really good question. And part of our work in Barbados, we started off with a very interesting model framework which allowed us to really understand and capture these delayed and nonlinear impacts of exceptionally wet and dry conditions and heat on dengue risk. But then when we moved to the implementation phase, we saw that this was quite demanding on the operational services because it required a whole new set of lags and leads for the climate information to be plugged into these models. So we started developing a more parsimonious approach to just really try and identify which are the most important hydrometeorological drivers. Can we just select the key drivers that could be picked out and to minimize any burden on an operational climate service center? So in our new approach, it's only one piece of climate information that is needed to be plugged into the model from a forecast. So we're trying to make things as efficient as possible and developing our tools using open source, reproducible code and methods so that these tools can be implemented within the routine climate services or the health services, depending. Each country has its own context, and sometimes these tools are operated from a climate or an environmental centre, where in other places it may be the Ministry of Health itself who might issue this kind of modelling outputs. So yeah, we're constantly trying to work on making our tools as parsimonious and efficient as possible.
Thank you so much, Rachel. Dr. Sundram, this considering that climate change is a global driver of vector-borne diseases expansion, but public health resources are often highly localized and limited. How can health systems design sustainable, context-specific adaptation strategies that balance short-term emergency responses with long-term resilience without exacerbating the health inequities between high income and the low income regions?
Maybe
is that to me?
Yes.
Yeah, go ahead, Bala.
Yeah, maybe I can answer a little on that. Basically, it's a good question because it's a systematic level question where you're looking at a central paradox of vector-borne diseases being driven by global climate change, but public health capacity is highly localized, uneven, and resource constrained. So yes, we have to weigh in that. But let's get the guiding principles right here, in which adaptation must not widen the gap between well-resourced and under-resourced settings. So we need to prioritize measures that yield immediate benefits and remain useful under future climate conditions. So simple things like mosquito proofing your housing, safe water access, health literacy, and building on the existing community structures, the norms, the beliefs. That's very important rather than importing a one-size-fits-all model. So that's very important. And emergency interventions, of course, we have our early warning systems, what Rachel mentioned, and Kathy has mentioned on the Wolbachia and integrated vector management. But that is also a very important component as well. And the long-term resilience, of course, we have whether it's infrastructure or health systems resilience. We get a lot of we want to climate-proof our healthcare facilities. That is something which we faced when a couple of years back when we had a massive flooding. So we need to have backup our flood-proof design and to maintain service delivery to the community. So that's another component. Yeah. Anybody can maybe add on. Yeah, thanks.
Anybody want to add something that is a very very important? And then so once even we know things are and then new tools and new techniques and things can come, but that ultimately the system should be able to flexible enough and then to have adapt it and implement it. So then with this thing, I want to ask this final question. So this is to Katie, that about Wolbachia. This have we seen any significant reduction in cases of dengue fever where the Wolbachia bacteria have been introduced, and how would you compare it to an area where the SIT program has been used?
Okay. So the answer is yes. We've seen sustained reductions in dengue cases in countries in Southeast Asia, in Indonesia, in Brazil, in Colombia, where Wolbachia has been introduced into mosquito populations, both in trial conditions and in long-term monitoring through disease surveillance systems. Compared to SIT programs, to my knowledge, there's not been efficacy trials with epidemiological endpoints of SIT programs yet to be able to measure that. The key difference is that SIT programs require the ongoing release of sterile male mosquitoes to suppress the mosquito population, just like you would with any mosquito reduction approach, whereas Wolbachia-based population replacement is introduced and then self-sustains in the mosquito population. So that's another key difference.
Okay, thank you so much, and thank you for your time and joining with us today and to enlighten a very important key area that the climate ready classrooms, the public health faculty development course. This is about the vector-borne disease, emerging risk, and intensifying threats. So there are a lot of particularly tropical countries. It's a burning issue, and then with this climate change and more risk, and then maybe health system has to spend a lot of money to provide the care services, and it is good to see how we can prevent and make that efficient way to handle this. So thank you for your very informative and then practical and experience talk. So I hope this was very helpful for our attendees today. And then so we hope to see you in a week time. Thank you so much. Have a nice day and good night. Thank Thank you.
Thank you so much.
Thank
Take
you,
care.
guys. Thank you so
Thank
much.
you.